So I'm Peter Martin, I'm the director of the Center for Biologics and Evaluation at the Food and Drug Administration. And what I could have talked to you about a lot of different things today, both cell and gene therapy. But what I'll talk to you about is what work we're doing to try to advance the development of gene therapies. And many of the principles that we're going to talk about for gene therapies actually have applicability to cell therapies as well. So in the next 20 minutes or so, I want to try to talk about some of our recent gene therapy approvals, talk about some challenges in product development, some of what we're trying to work with others to overcome in terms of challenges, and talk about some of the resources that we have. Much of this is not going to be new to you. It's stuff you've heard, but we'll try to give it my particular spin. So in case you've been under a rock for the past couple of years and you haven't realized it, there are two CAR T-cell therapies that are currently approved in the United States, tisagenlecleucel and axicabtagene. Those now cover ALL and acute lymphoid leukemia and non-Hodgkin's lymphoma, and the other for non-Hodgkin's lymphoma. There are a number of other CAR T-cells which are coming up close to approval, and we'll see where those go. We also now have an ocular directly administered gene therapy, voretigene, for a hereditary retinal dystrophy, which was approved using a very nifty endpoint called the multiluminance mobility test. If anyone wants to see kind of an interesting endpoint that actually is patient-focused, that was used for this particular approval. And it was very nicely developed by the company in collaboration with some input from the agency. And so you can get online and see this. Basically, it's a test where people navigate a maze under different amounts of light, and they were able to show actually an improvement in that. That was that approval. And we also have now, more recently, a systemic directly administered gene therapy, anasemnogene, which is for spinal muscular atrophy, a disease which is really quite terrible in its most severe form, where it basically takes children's lives within the first few years of life. These are children who, usually by about a year of age, end up on ventilators, unable to move, and then shortly thereafter tend to die. This was an example of the kind of clinical data that I like as a non-statistician, because you don't need a statistician to talk about statistical significance. Basically, one could look at natural history and look at a disability score and show that by a certain number of months of age, no one had surpassed a certain milestone, and after the gene therapy was administered, and you can see 15 patients on that lower left. These are patients now who were not on ventilators, who would have been expected to be on ventilators. And the child on the right here is a child who had been treated early on, who basically appears normal, who, if she had not been treated, it's pretty certain would have either been on a ventilator, essentially on last stages of dying from this disease, or not with us. And instead, she was running around. She actually was brought down to testify to a congressional meeting and was running around, and she really got the senators very riled up. She was very cute. So what's happened? The fact that you're all here and the fact that this meeting has needed more and more space basically shows that success brings interest. There's clearly increased activity in gene therapy or cell-based gene therapies. When I say gene therapy, I'm talking about vectored gene therapies as well as cell-based therapies that use a gene therapy vector in their manufacture. And you can see that between 2017 and 2018, we had roughly a doubling in the number of investigational new drug applications, now with over 800 active investigational new drug applications on file at FDA. That means ones where we're actually actively having correspondence with the sponsors. That's pretty impressive. This year, we're well on our way to probably exceed those numbers from 2018, probably by about 50%. I can't give you the exact number yet, but for calendar year 2019, this growth continues. What's that going to turn into? I think you've already heard at this meeting that it's likely that we're going to start to see more and more products reach market. This is not FDA data, this is MIT data, just showing that they're predicting by 2030 there'll be 40 to 60 product launches with more than 500,000 individuals treated. Could it be more? Could it be less? Of course it could. And so there's some error inherent in this, but we are on this essentially very steep portion of a growth curve. A lot of this has been facilitated by the fact that the field has settled, for the most part, on two major systems, lentiviral vectors and AAV vectors. That's not to diss any other vector systems or non-viral vector-mediated gene therapy. It's just to say that a lot of the advances, for instance, for the CAR T cells have come through the use of lentiviral vectors, and for directly administered gene therapies through adeno-associated viral vectors. And so I'm going to just focus in a little bit on adeno-associated viral vectors, just because it's a good example that shows some of the challenges here. And again, I'm going to use this as kind of a case study that might be applicable to some of the other products in this space. So currently, there are a number of challenges in the development of AAV gene therapies. One has to deal with this fact that there can be pre-existing immunity to adeno-associated virus. It's something that we're exposed to, and particularly if you have a disease that's not common and you're going to be treating this with an adeno-associated virus, you want to make sure that the treatment that you're going to be giving to it is going to be effective. So you have to try to deal with pre-existing immunity. There's this issue that we still have to figure out long-term safety and efficacy for these products. No one really knows exactly what's going to happen 10 years down the line, 15 years down the line to those children who have been treated with the SMA1 product. Will be interesting to see that. We're going to have to figure out how to most efficiently put forward clinical development, because we're now into a new mode where our typical paradigm of doing randomized clinical trials in hundreds of patients, this does not fit this model at all. And we're also going to have to deal with probably one of the most significant things is how we're going to manufacture these things. And I'm going to save the bulk of my time to talk about that. So just to talk a moment about this issue of pre-existing immunity to adeno-associated virus, I mean, it can preclude effectiveness, and developing immunity, once you've received it, can preclude redosing patients with the therapy, which could be an issue. But there are strategies that are under exploration, and as someone who's a hematologist-oncologist by training, we know how to use immunosuppressive regimens. We know how to use technologies such as apheresis to remove antibodies. So there may be ways of getting around this by brute force. There may be more elegant ways of getting around this by using vectors that have been had their surface characteristics changed so that they don't elicit the kind of immunity that wild-type AAVs do. And so that may be another way around this. And then another way may be just to move away from AAV entirely to other viral and non-viral vectors. That's probably a little further off. In the area of assessing long-term safety and efficacy, there are right now conventional protocols that are being used to extend follow-up of patients who have been treated with gene therapies. But also now people are starting to think more about using patient registries curated by entities other than the sponsors. Those are maybe by advocacy groups or others. We're starting to use real-world data. And I'm a little hesitant to use real-world data is not a complete panacea for everything in drug development. Although CEOs sometimes drink the Kool-Aid for real-world data fully. But it is potentially powerful in this area, because as more and more people are treated and as we have better and better electronic health records, it may be very helpful in following people, particularly because some of the electronic health records and these claims databases that are being set up now are designed to be able to follow people across different insurance companies. That is one of the problems here, which is that in the United States, can't speak for Europe, but where it's a little different, but in the United States, the average person changes health care insurers every 3 years. And maybe even more than that now, but that was, as of a few years ago, that was the number. So we need to be able to follow people long-term with these, and hopefully we'll have systems that will do that. Towards implementing efficient clinical development, So first of all, I think stakeholder engagement is something that's very important in this area, because when we're developing things, we want to develop them to deal with the issues that people really care about. Now, you might say it's just the life or death issues, but for many of the rare genetic diseases, it's not just the life and death issues. Unless you're dealing with some of the things that make living life unpleasant, like feeling horribly fatigued every day, or being unable to walk more than a few steps, you're not actually really dealing with the issue. So stakeholder engagement early on is something that's probably very important. We have issued guidance recently on patient-focused drug development. It's a good place to go for some advice in that area. We're also thinking about different types of clinical trial designs which might help in this area because, as I said, the conventional large randomized clinical trial isn't going to be useful here. We're willing to consider Bayesian designs and complex, innovative trial designs where you try to minimize the number of patients treated in order to come to a conclusion about whether the therapy is safe and effective. We also will make use of all of our expedited development programs. Some of those you're aware of. I'll mention them in a moment. And I would also stress early discussions with regulatory authorities. In order to help people figure out how to navigate this area, we've issued a suite of gene therapy guidance documents. The draft guidances were issued in July of 2018. We got back a lot of comments to these through our comment period. And probably before the end of this year, you'll see the final versions of these incorporating those comments come out. We'll also probably have some other guidance coming out in the not too distant future. Things on, for instance, orphan sameness to understand whether two gene therapy products are the same or different from one another, if they have the same insert but different vectors, that type of an issue. So our expedited development programs, for a while in the United States, people are familiar with fast track, priority review, accelerated approval. Breakthrough therapy designation was put in place in 2012. It's being used pretty widely. And in 2016, at the end of the year, we had the Regenerative Medicine Advanced Therapy pathway put in place by the 21st Century Cures Act. These are all programs for serious diseases. The Regenerative Medicine Advanced Therapy designation, which is very much like breakthrough tailored to cell and gene therapy products. It's a very similar program to breakthrough therapy. The bar for actually getting the designation is slightly lower than for breakthrough therapy designation. And the number of ways of fulfilling post-marketing commitments, if one gets an accelerated approval, is expanded with the RMAT designation. But there has been a lot of interest in this. And we're finally seeing products that have come all the way up through this process. And so actually, I look forward to the day when I can tell you what has come through the process to market. But to date, we've had, as of August 31st, 2019, we've had 115 requests. 44 of these were granted. 61 denied. Most of the denials had to do with the fact that either people were studying a product other than the one that they had actually submitted a request for, or that they didn't submit enough clinical data. But that being said, the numbers here, 44 out of 115 requests, those are pretty close to the breakthrough therapy percentages in terms of what's granted. So I think not doing too badly there. In terms of coming in early to the agency, we have the Interact program. This is essentially replaced our pre-IND meeting, which nobody understood because it sounded like pre-IND meeting, which is actually a formal regulatory meeting. This is essentially an informal meeting with non-binding regulatory advice where you can come in and talk about a product that you're developing. And if you have questions about its preclinical development, its manufacturing, or for that matter, its clinical development program, one can have a non-binding discussion about what that might look like. There's a link here, but you don't really need the link. If you type in Interact and FDA or Interact and CBER to your browser, it comes up pretty quickly. And there's a pretty step-by-step guide of how to send in the information for this. It's not very complicated. Probably this is something that's like one of those term papers that you can do the night before you need to turn it in. For CATS meetings, we've also have realized that for many of the people in this room, you don't have a specific product, but you have a platform that you're going to manufacture a cell therapy or a gene therapy on. And you might like to understand how the agency would think about things manufactured off of that platform. And so we've now developed an advanced technology team, which is analogous to the emerging technology team at the Center for Drugs, which is really to talk about platform technologies. And that allows a pretty flexible, again, non-binding discussion before a regulatory submission is filed. And again, this can be, you can find out how to submit for this. Very similar, informal submission, not a lot of information required, just enough for us to understand what you're coming in to talk to us about. But I want to spend the last number of minutes here on challenges in manufacturing gene therapy products, because this is really, to me, one of the real areas where we still have a ways to go. We still have issues with process scale-up, which is not yet reproducible from clinical trial to commercial size lots. I should be fair about this. There are some people in the audience, there are some companies that have got this down, but it's not uniform yet. There are clearly limitations on information exchange from commercial confidential trade secret perspective that are potentially limiting the evolution in manufacturing technology in this area. And unfortunately, for better or worse, the production of the viral vectors that are in our current generation of cell lines is relatively inefficient and it's costly. And that's leading to some issues. And it's leading to a reality that we just have to all accept. Right now, gene therapy is really, I'm sorry, this is my coffee analogy because it's someday when they throw me out of the federal government, I'll go work for Starbucks and I can deal with tall, grande, and vente. I did not put Trente. That's the really large cup they have that you can ask for. But the sweet spot here is the grande size. It's why they don't even put the tall or the venti prices on the wall anymore. Because we're good right now at thinking about making gene therapies for somewhere more than, how many hundreds, I can't tell you, but hundreds to tens thousand patients, we can imagine making gene therapies for those kinds of populations. If one were to think about, if I had a gene therapy that could address heart disease in the United States and bring down the risk of middle-aged person's heart disease by half, and it was safe and effective, that's great, but we couldn't make it right now at a scale if it had to be given systemically. So patient populations more than 10,000, is it exactly 10,000? I can't say 20,000, but large patient populations, I don't think we're viable yet in terms of the technology with scale up or scale out. But I'm going to focus the rest of my talk on a place where I think we really are going to have to get used to, because the scale up to the 10,000 or more is true. It's like the monoclonal analogy. You have to think about how you're going to do things. But the problem with the small gene therapies, these small numbers of doses that are going to be needed between 1 and 100, is that's where a lot of rare diseases sit. And ultimately, as we think forward towards where we're going to go with genome editing, when you start slicing and dicing even more common diseases by putting in different guides, that's where you're going to also end up for some of the gene therapies there. That's also potentially where you might end up with gene therapies using antisense technologies to deal with different mutations as well. So we're going to end up in a situation where the problems are a little bit different than scale up and scale out. The problems are more analogous to what one has when you go to a printer and ask for wedding invitations. There, whether you ask for 10 or 50 invitations or 200 or 500 invitations, the setup cost is the same. And so the price is not all of that different. So this is a problem in this area because we're going to need to be working here, and yet we don't have commercial viability. And unfortunately, it's really important for people with ultra-rare disorders, because if you think of it, there are really hundreds to thousands of disorders that affect 1 to 100 individuals that could be addressed with gene therapy. And as I said, genome editing may drive more towards this area. So what are we trying to do at our center? Well, for the larger scale problem, for the scale up, like many of you in the audience, I've already heard some discussion of people who are working to try to make better cell lines, better production technologies. That's something that we find very important, and so we're trying to work towards making improved producer cell lines and bioprocesses, manufacturing technologies that might allow higher yields. But we're also trying to address the manufacturing needs for ultra-rare gene therapy products in kind of a pre-competitive space. One of our concerns is that even if we address these right now, how are we going to make these things so that people will have access for them in the near term? I just want to caveat what I'm about to say with the following. I have no doubt that 10 to 15 years from now, there will be a business model which will take care of this. It may be a business model where it looks more like a device that produces different gene therapies in small amounts. And so there will be commercially viable strategies there. Those of you who want to go there, please do. No charge for the suggestion. But for right now, we have this need for access. And if you're the parent of a child who has a rare genetic disease that's going to kill them in the first years of life, you want the technology now, not years from now. So what we've been thinking about is whether we need something like a consortium, a public-private partnership that can address the end-to-end development of gene therapies for these very small niche disorders, so that when you need 20 doses a year, which is not going to be, no one at this point is going to get excited about making those for a rare disorder, that there can be access to these. Our reason for trying to think about this consortium is the idea is if you have a consortium where you leverage what FDA is willing to do at this point, which is we're willing to start to think about using master files for a vector, master files for a manufacturing process. You could potentially take what's currently taking 4 to 8 years from concept to patient results and half the amount of time, which, again, if you're the parent of a child with a rare disease, or for that matter, if you're anybody, you can understand what the benefit of that would be. So we want to see this technology be more accessible to academic innovators, applicable to a broad range of diseases. We want to try to streamline the manufacturing information needed. That's going to be good for everyone because this will actually benefit the entire field. If we can figure out the regulatory pathway to facilitate streamlined data submission, a streamlined process for these kind of new generation of individualized or bespoke therapies, it will make a difference. I think ultimately the process development that we will have as we address this kind of small batch gene therapy may actually end up rubbing off onto the larger products and help us develop better cell lines, better technologies. The throughput itself will help, right? You do something a lot and you learn a lot through it. And ultimately, we hope to bring these therapies to individuals in need sooner. I think that's what everyone comes to these meetings. Ultimately, that's our goal. So we're very committed to advancing the development of gene therapies for patient populations of all sizes. Sorry for the cell therapy people here. We also care about cell therapy deeply. But I use this as given a half hour of time, I wanted to try to be more specific about something. We want to encourage innovative product development, clinical trial designs, and we actually realize we have to collaborate here. We understand that we don't have all the answers. We understand that this is a field in which we have to essentially hold on to our hats because things are changing so quickly. So we'll look forward to working with everyone. And I think I have about 3 minutes and 30 seconds for questions now. Thank you.
Sorry, I missed you.
That's okay.
Thank you.
This is Michael Werner.
Yeah. Thanks, Peter. And just for those of you who've been to this meeting before, you know that FDA has attended this meeting and provided candid remarks for the last 10 years or so. And it's really something that ARM is very proud of, is the relationship and collaboration that we have with the FDA, and thanks, Peter, for all of your leadership that has helped make that happen, and we've all seen the benefits for patients so far, and many more to come. So we've got time for a question or two. So, is that Bob?
Any thoughts that you have on what you'd like to see accomplished?
Yeah, I think whether or not it comes with PDUFA or with the PDUFA Christmas tree and the reauthorization, my biggest concern is that although we're working to staff up for actual submissions in terms of BLA submissions, license applications, we need to deal with that as part of the next user fee agreement. But the most important thing that I think we need to think about right now is how we're going to provide the early advice, which is we're beginning to become overwhelmed with people that want early advice about development programs, development platforms. And because that's not really taken into account in how our staffing is, it's putting a strain on staffing. So I'm the first to admit that people, when they want advice, they don't want to wait several months from FDA to get back to them. And so we feel like we need to staff up in this area so we can get back to people in a more timely manner. My goal is if a company comes in, we'd like to get back to them within 30 days, not 120 days. And that's where we are right now in terms of averages. So we'll need to staff up in this area, and that's, I think, a conversation we'll need to have.
Okay, last question.
I'd like to make a comment, and then I'd like to ask a question. So on the comment side, we've been in this industry for about 20 years, and we were founded based on the work of Jude Samolsky, who was the first to clone AAV for therapeutic purposes. So we've had some time to think about some of these issues. With regards to the ultra-rare indications, our solution was to have a sister nonprofit organization that focused on developing therapeutics without the pressure of getting a profit. And we integrated some technologies, such as doggy bone technologies, an alternative to DNA plasmid because it's quicker and cheaper to manufacture, and then things like wave bioreactors, which allow for smaller batches. So I think there's ways to do it, even within our construct, but I'm really excited about the public-private partnership. And then I have one question for you. And that is, given the, we have, I guess, legislation for orphan drug development, we have tax incentives. Why isn't there legislation for ultra-rare indications where there's another set of tax incentives for for-profit organization and for nonprofits to make investments?
Great question. We don't lobby Congress at FDA, but you may. And so please feel free to bring that. I think it's a wonderful question that's come up. And I think in some ways this is what living in this area has been like, because I think 3 years ago we wouldn't really have been having this conversation because people were just grappling with, could we do this for some disease? Now that we're realizing that this is potentially an accessible field for an incredible realm of diseases, this becomes a much more viable question. So be my guest. Thank you so much. Thanks.